In this video, I will talk about a class of macromolecules known as nucleic acids. Nucleic acids are polymers built from the monomers or simple subunits of different nucleotides. For any given nucleic acid, there is typically only 4 different monomers or 4 different nucleotides that are used to build the polymer. 2 examples of nucleic acid polymers would be DNA, or deoxyribose nucleic acid, and RNA, ribose nucleic acid. The main function of these polymers is to store or transmit genetic information. Let's take a closer look at some of the monomers that form these nucleic acids. These monomers are known as nucleotides, and there exist several different types of nucleotides that can be used in building a nucleic acid. However, these nucleotides do have a common structure. They will all have a phosphate group attached to the so-called 5' carbon of a sugar group, usually a 5-carbon sugar like a ribose. And this sugar will also have attached to it a structure known as a nitrogenous base. The 2 different types of nucleic acids I mentioned, ribose nucleic acid or RNA and deoxyribose nucleic acid DNA, differ in what is attached to the 2' carbon in the sugar. Essentially, DNA is missing an oxygen in that place or in that spot, and thus it's called deoxyribose. The nitrogenous base of different nucleotides is what defines a nucleotide, or it's the feature of the nucleotides that are different from each other. Here's a view of some of the different nucleotides you'll find in RNA and DNA, or in particular, the different nitrogenous bases on those nucleotides. You'll notice that for adenine, guanine, and cytosine, these will be present in both RNA and DNA, where in DNA you'll have thymine. In place of that nucleotide, you'll have uracil in RNA. They have very similar chemical structures. Nucleotides are linked together through dehydration synthesis along their common features. The phosphate group of one nucleotide attached to the 5' carbon will form a phosphodiester bond with the hydroxyl on the 3' carbon of the other nucleotide. So we're going to link the sugar of one nucleotide to the phosphate of the other. Notice how this linkage will occur irrespective of what type of nitrogenous base is involved. So the nucleotides shown here could be, as depicted, linking uracil on top with adenine on the bottom. Could very well have been cytosine on the top and guanine on the bottom. Here's another view of the type of linkage that's going to occur. You can notice that a polymer of nucleotides, or a nucleic acid, will have a direction. One end will have an exposed phosphate. This is sometimes referred to as the 5' end, after the carbon to which that phosphate is attached. The other end of the molecule has an exposed hydroxyl on the sugar in the 3' carbon. This is usually referred to as the 3' end of the molecule. New nucleotides are added to the chain, or to the polymer, onto the 3' end. This actually occurs because incoming nucleotides carry more than one phosphate. The removal of the other 2 phosphates helps to provide energy or create energetically favorable circumstances for addition of the other nucleotide. Even though nucleotides might seem rather boring and straightforward, since they're linked together in the same way, it's the order in which the nucleotides are linked and the presence or the order of which nitrogenous bases are where along that chain that really carries the function of nucleotides or of nucleic acids. The sequence in which these nitrogenous bases are found represents a form of information, just the same way that the sequence of letters on this slide represents information. If I was to scramble the letters in one of these sentences, it would lose all meaning. So you can think about letters in an alphabet or a digital code. For example, a barcode, probably a little more analogous to nucleic acids since there's only 4 different choices. Just as for a barcode you have actually only 2 choices, a white or a black stripe. Nucleic acid information can be very complex. Here is a nucleic acid molecule, a molecule of DNA, represented by abbreviating the nucleotides in the order in which they're found. So for example, this particular molecule of DNA starts with adenine, thymine, guanine, then adenine, represented by A, T, G, A, and so forth. Now, this might look like nonsense or gibberish, but the cell has ways of interpreting this information, and actually this particular set of information is something meaningful. This is a gene which will impart to whatever living organism has it, at least under the right conditions or in the right context, a green color under UV light. It's the gene or the protein coding region for the so-called green fluorescent protein gene. The way in which this information is decoded and processed is complex. But in essence, the information found in a molecule of DNA represents a recipe for how to make one or more proteins. This recipe is copied into a working form and then processed or decoded or translated by a molecular machine in the cell known as the ribosome. This will form a protein with a particular amino acid sequence, which then will cause that protein to fold into a particular shape. Just the same way that a recipe contains a particular set of instructions for making a particular meal out of other materials. Of course, you don't use the paper the recipe was printed on in the final meal.